And our vision is that we would like to be as one of the very only companies worldwide who is busy in the R&D field for plasma fractionation. We would like to bring in the next years 5 drugs at least to the public so that we can improve millions of patient lives. And therefore, Prive Pharma was founded in 2015, starting with fractionation and chromatographic issues. And just now in 2023, we are going to build up a GMP facility where we can start to produce our own material and our own drugs so that we can go even until phase 2-3 into clinical trials. We are just now currently 30 people, have a lot of space, 400 square meters lab space, a dozen additional office space. And as I said, we are going just now to get a 600 square meter GMP office so that we can start also the production over there. What we can offer, that's the very classical things we are doing very often for the big fractionators, life cycle management studies. We are doing also process development, and we are developing also analytical assays for quantifying, for qualifying in terms of potency and stuff like this, so that the big fractionators can do the outsourcing of their R&D part into our company. We are doing our own innovations, and we call it Medical Innovation Platform, and we have just now between 60 and 70 different new plasma proteins on our list, which we're going to bring step by step into progress. And what I said just now, we are planning to have the manufacturing available. Our current limiting space is about 50 liters, and the new space we're going to allow us to work up with volumes of 500 liters, and the final extension we're going to need up to 2,000 liter plasma fractionation size, so that will be sufficient for the majority of all products which should going to go into clinical trials and even sometimes onto market. Plasma itself, it's a quite interesting topic. You know, when I started studying medicine, there was a list of about 180 different plasma proteins. And just now we know that we have more than 10,000 different plasma proteins, and currently 11 or maximum 18 of them are used as commercial products. All the other proteins are wasted, and that's something what I feel, or even as a physician, is unethical, that we are leaving our donors sitting on the apheresis procedure for 1 hour, and then we are using only 11 of these proteins. So that's something what I feel is our task, that we have to find more indication for this. And we have to keep in mind that there are some trends ongoing. So there is an increasing use of those plasma products in acquired deficiency in the Northern Hemisphere. So I'm going to come up with some examples later on. But we have a decreasing use in genetic deficiencies. And as Ranjit said already, we are facing the trend to gene therapies. So these gene therapies are just now present already for hemophilia A, hemophilia B, that's a little bit away, and in some cases of immunodeficiencies. And so the market will go into acquired deficiencies. There's just a simple example, something what we are called secondary immunodeficiencies or acquired immunodeficiencies, or even iatrogenic immunodeficiencies. You know, we are treating a lot of leukemia patients with anti-CD22 antibodies. These antibodies were going to kill the B cells until no B cell is left anymore. And the result is a classical immunodeficiency, not in the toddlers and the newborns, but in adult patients. And they are facing the same symptoms as the classical immunodeficiencies. And this market we're going to grow and grow and grow and reach earlier or later the same size as the market for primary immunodeficiencies. That's something what we have to keep in mind, that this market is everywhere present. And the more we are going into those modern monoclonal antibody-based therapies, the more we have to keep those secondary immunodeficiencies in mind. And these antibodies were not only used currently in leukemias, they were also used to treat severe autoimmune diseases like lupus erythematosus, like sclerodermia and stuff like this, or even severe neurological inflammations. So that will be something where we have to be, as physicians, aware that we didn't bring our patients too deep into the immunodeficiencies. We know already that from a couple of therapies, deficiency in the coagulation pathway are present. So there is always some papers about acquired factor VIII deficiencies. That's something where we have to aware as well. And we're going to have an increased use of inhibitors. 83 is already well known, and which is used in the case of sepsis, and protein C as well. And the protein C is quite an interesting protein. When you're looking on the gene maps, you're going to find up to 95 mutations on this protein. And especially in the Asian room, a lot of patients suffering from protein C deficiencies, which becomes only obvious when especially the young patients get infections with gram-positive bacteria. So all of those are going to require the treatment with plasma proteins. And we have another trend, which is quite important. When I started with the plasma fractionation, and when I come first in this business, the fractionation capacity was about 20 million liters per year in 1992. We have just now worldwide a plasma fractionation capacity of 45 million liters. And we are still short on plasma because if plasma is not an unlimited source, it starts with the plasmapheresis, and therefore we need to have donors and all those facilities for plasma donation. So we can expect that even when we are filling the whole fractionation capacity, that it would not supply the whole world market with plasma products. And that means for especially Asia and even Latin America, that they have to go for their own fractionation and for their own plasmapheresis. I'm going to go later on into some details. So there is for sure some trend for the plasma fractionators, which is quite important. They can increase the business by improving the yields. Just a simple example again, when I started in 92, the yield for immunoglobulin was between 27 and 28%. And we are just now in much higher regions. We are just now getting up to 80% of the total immunoglobulin out of the plasma. So that's a huge increase, but immunoglobulins are still short. And that's quite simple. On one hand side, we are going for new indications. And on the other side, I said this already, we are facing those new developed secondary immunodeficiencies. And a simple example also here, the use of immunoglobulin in post-polio syndrome. That's something which is described, but we have still many post-polio patients. These are patients which had the polio infection 20, 30, 40 years ago, and they are suffering from this post-polio syndrome when they are getting 60 or 70 years old. Or we are using albumin in intoxications in septic patients and stuff like this. So for sure, it's important for the plasma fractionator to extend the business by going into new indication, which can be on one side the classical therapeutic areas as they are known for plasma fractionators, but they can be also, especially when we are thinking about the new proteins, outside the classical indications for plasma fractionators like osteoporosis, or stroke, or chronic inflammation. That will be something what we have to face when we are going into detail. And therefore, Privi Pharma developed this MIP platform. These are our list of proteins which can have an impact on the next years. And so we are searching for plasma, potential plasma-derived products in the patient data. We are doing the patient data by clinical trials because we are located just in the middle of the Mannheim University Hospital, so that we can have access after ethic votes to patient material and can check them for different parameters. For sure, we are searching patents in literature. We are observing marketing data and stuff like this. And as you can see here, we have currently many therapeutic areas in focus. starting with metabolic diseases, including osteoporosis. We are focused on infectious diseases. We have an eye also on the transplantation immunology, on autoimmune diseases in like rheumatology or neurological diseases. Hematology and hemostasis is quite important for us, surgery as well. And we are also looking for this complete outside living therapeutic areas like ophthalmology and anti-aging. And that's a kind of basic scheme what we are doing. So on one side, we are on the discovery field to identify out of this 10,000 different proteins, potential other proteins which can play an important role in the pathogenesis of a disease. Therefore, we have our medical expert and doing the clinical and diagnostic studies by ourselves, defining the medical use case. And the implementation, that's a part which is just now very interesting for us. We have to be able to produce the material. So it means that we are developing methods which are easy to handle and easy to use. We are validating those methods so that they can be upscaled up to 1,000 or 2,000 liter scale, and that they can be used by the fractionators and even in small scale fractionation. We are doing the preclinical proof of concept as well, either in the cell culture based assays or even up in animals, so that we have the whole spectrum of data which are necessary for licensing a new project available. Just a short review on the plasma fractionation industry. And as you can see here, the majority of plasma fractionators is based on the Northern Hemisphere. And these are the big players here. Everyone knows CSL, Behring, Grifols, Octapharma, Kedron, Biotest. The others are the smaller ones. And as you see here, the majority of them is based either in North America or in Europe. And the question is how we can support patients in the Asian region. There are living many peoples, and therefore a solution has to be present. Looking a little bit more into detail, that's something which will be quite interesting. As I said already, we are using currently 11 to 18 proteins out of the 10,000. And as you see here from the market growth, it's still increasing. We're just now with 27 billion turnover. And facing this year in earlier times, in the end of the last century, the business was driven by albumin and by the coagulation factors. And just now we have not the opposite. We have the major driving factor. These are immunoglobulins and albumin again. It comes back, and the coagulation factors are just now replaced either by gene therapy or by recombinant coagulation factors. That's not the major business anymore. But as we're going to hear later on, there are still some indications in surgical situations or in trauma situations where we need to replace plasma at all to get the whole composition of coagulation factors available again for the patient in case of severe bleedings or stuff like this. The classical fractionation, that's the one which is described by Cohen and modified by Kistler and Nitschmann. You know, at the beginning, we have this cryoprecipitate, which is the base for factor 8 and von Willebrand, fibrinogen, and it's also the base for the C1 inhibitor and the proton complex. The precipitate 2,3 is the base for the IgG purification. That's FR1 antitrypsin, that's also quite important. And the very last part is albumin, which comes out of this. All of this required, as you can see here from the temperatures, the pH, and the alcohol content, a lot of fractionation equipment. Summarizing like this, so we're starting with the collections, pooling, and as I have learned, large pools contains up fractionation capacity per day is between 3,000 and 6,000 liters per fractionation plant. And I'm going to switch just now my camera off because I have seen that the web is a little bit unstable. Then we are going for the fractionation, cutting into different pastes, protein purification, mainly by chromatographic methods, and then the formulation. That's a classical method, which looks and which works in big tanks with huge amounts of alcohol which have to be removed, and all those stuff. And so we can summarize this process as here the donor is the key in this whole process, and it's very important that the donor is coming back latest after 1 week, which I feel is quite interesting. Plasma has to be tested and virus and frozen. Then it goes into the inventory hold, and the different fractionation processes are going for the cryoprecipitate for the fraction 1, 2, 3 and fraction 5, so that you get the main coagulation factors here and IVIG and albumin over there in this process. And it looks like this. I have seen from some fractionators the pictures. This picture is taken from Baxter. A lot of tubing, a lot of vessels, stainless steel, very expensive. And even when we are going one step ahead in the lab scale, we are using those columns for the purification and polishing steps. The bigger ones are looking like this, and the very big one are this one. So you can imagine that it's quite expensive and quite complicated. Despite of all this vessels for reactions, you need to have a freeze dryer, you need to have buffer preparations and mixes, you need to have diafiltration, ultrafiltration, chromatographic systems, and even sometimes a centrifuge when the centrifugation cannot be replaced by filtration. So the last step is always, or the step can be included as well, the viral inactivation. Here I used simply one example, the nanofiltration. You know, there are different viral inactivation methods accepted by the authorities. It could be low pH, it could be heat treatment, it could be SD treatment, or here in this example, the nanofiltration. That's a quite established method. But summarizing all this, we have to start with the plasma sourcing. And there is, for my opinion, a quite clear conclusion. As I said already, we have currently 45 million liters available. 80% of this plasma is coming from US, 20% from Europe. And when you're looking for the European map, we're going to find that only Germany, Hungary, Austria, and the Czech Republic are self-sufficient. These are these 4 countries which are here, dark or dark gray. All the other countries in Europe are still not self-sufficient. So the conclusion is that it could be not expected that larger quantities of plasma products can be exported to Asia. And the major conclusion is Asia needs to get self-sufficient. And to being self-sufficient, Hanji, you know this quite well, using recovered plasma, that's one way. And I can say, don't waste any mil of this recovered plasma. But the introduction of plasmapheresis, that's an urgent need when you would like to be self-sufficient within a short time. So we have a lot of experience in building up plasma centers and plasma equipments, even testing equipments. And so the conclusion for me is blood and plasma services needs to establish locally with a huge extent. So we are expecting in Europe that we need to have at least one plasma center, or one, let's call it blood service center, because of the blood is needed, the platelets are needed, and the plasma as well, per 100,000 or 150,000 inhabitants to be self-sufficient. So to do this, we've learned from Europe and the studies we have done that the donor compensation is necessary. And you can have a look into the PPDA documents where this map is coming from, that the donor compensation is really urgent need. Don't pay the donor, but compensate the time the donor needs for one plasmapheresis. That's normally 1 hour of time where the donor is sitting in a blood service center to donate his plasma. Interesting is that we have different rules worldwide. In the top of this table, you're going to find the US rules. They can donate, depending on the body weight, between 690 and 880 mils. You can calculate this without anticoagulants, and they can donate up to 104 times. We know already that this is too much, and from the Sidler study we have done between 1999 and 2003, and which was published, we've learned that the IgG of the donor is limiting. So it's very important for the donor safety that the donors are checked in regular levels for their IgG level. And if the IgG level becomes too low, we have to stop and reduce the donation frequency. That's the Austrian variant. They have different volumes to donate. And there it's fixed without any anticoagulant, and the Austrian inhabitants can donate up to 50 times. These are the simpler conditions, and these are the only published conditions, so you can call it validated. And since 2017, we have those rules here in Germany, which are a little bit adjusted to the simpler conditions. Simpler stands for Study for Intensified Plasmapheresis. Body weight between 50 and 60 kilograms, they can donate 650 mil, 70 kilograms, 750, and larger than 7 kilogram body weight, they can donate up to 850 mil. And the number of donations is regulated with currently 60 donations per year. That's quite fine, but finally we have to say that we should gonna find, at least in Europe and for other countries as well, a kind of general regulations which keeps on one hand side the donor safety in mind and keeps on the other side the product safety and the product quality in mind. Because of, as we know already from the US plasma, one of the problems with the US plasma is that the IgG content is between 10 and 12% lower than in European plasma. So the high donation frequency is not always good for the whole fractionation process. Here is one overview as it is run in different European countries. Sometimes they need to have a physician present. Sometimes it's not necessary. The regulations are described here, how many donations per donor and per year, and the volumes are described here, and it's kind of how to keep this all in mind. So it means at least that even the government and the regulatory bodies have to be involved when it's up to the regulation of the plasma donation in a respective country. Plasma has to be tested as well. There are some general rules from the PPTA. That's the IQPP Qualified Donor Standard. I don't go into detail here. And the QCL program, which is important for all plasma fractionators, but something has to be kept in mind. We are testing serology and clinical chemistry, and some of them is done for the donor safety. It means measuring the hemoglobin at each donation and total protein and IgG latest at every 5th donation. That was one of the major conclusions out of the Zippler trial, that we have to check the IgG latest at every 5th donation. Otherwise, we are bringing the donor into risk for infections. So in the hemogram, we have in Germany, not for all, but a little bit gender dependent, a hemoglobin level of about 125. The total protein is quite important, because if total protein is going too low, the donor is on risk. But we can expect that the protein loss by an apheresis is compensated latest after 48 hours. And just now the big but, IgG, the donor has to have at least 6 grams per liter in his plasma. And if you have once an IgG loss, you have to keep in mind that it took between 2 and 4 weeks until the donor is back to normal levels. And therefore, in the plasma centers where I was responsible for, we decided to let donors with IgG levels between 6 and 8 grams per liter donate only twice a month. Donors with IgG levels between 8 and 10 grams per liter, they can donate once a week. And donors with levels higher than 10 grams per liter, they can even donate twice a week. The other part, serology, it's important for the product safety. So we have to test for anti-HIV 1 and 2, anti-HCV, hepatitis B surface antigen, and in some areas, the Lewis testing is still required as a surrogate for promiscuity. That's quite normal. Some didn't ask this anymore, but it's about 50-50 currently. And NAT testing has to be carried out also at each donation. And the QSEAL program is requirement to test for HIV, at least with a dual target NAT, hepatitis B, hepatitis C, hepatitis A, and parvo-B19. And currently in discussion is the hepatitis E, which doesn't play a major role in the plasma donation. There are currently no infections described with this virus by plasma proteins. So the question is how to get more plasma in the Asian room. And there is one idea which we called chairside fractionation, which can allow to pick up the IgG directly during the apheresis procedure, put it on a cartridge and put this cartridge onto an inventory hold so that the fractionation takes part as everywhere. But this procedure avoids the shipment of huge amounts of water. That can be done also for other, but when we are talking about normal plasma donations, then it's quite important to keep the donor in the plasma center and make sure that the donor is coming as often as possible in medical view, so that the donor safety is not impacted, but often enough so that also the product safety is kept in mind. You know, we have this quarantine for 4 to 6 months, depending on the presence of NAT testing. And therefore, it's quite important to keep and to bind the donor into a center, and therefore programs for keeping the donation frequency as high as possible are needed. Some examples, what Previe Pharma has developed in the last years. We have one product which is called Previe 0202. This product can be used for deep vein thrombosis. You know, deep vein thrombosis is always a risk to end with a lung embolism. It's not that high present, but if you have once this problem, the patients are on really high risk to die. The risk to die is at about 15%. Lab diagnosis, that's quite clear. And what is currently available, a therapy that's mostly heparin or commonly known blood thinners as vitamin K antagonists or factor 10A inhibitors. But at least you can counter therapeutic approaches only as prophylaxis for the next thrombotic event. Now that's a huge market, and we have shown that these products can be used probably also in myocardial infarctions, lung infarctions, pulmonary embolisms, and stuff like this. Here we check the levels of those proteins in different groups of patients. In the controls, we have a level between 80 and 150%. In the acute kidney failure, about 40% of all patients have levels below the normal level. In DIC patients as well, in septic patients ditto. And as you can see in coronary heart disease patients, the majority of patients has decreased values. So a substitution can be probably quite helpful. We showed this in respective animal models. So we occluded the vena cava inferior by ligation, waited until thrombosis formed, and then we applied our Previe 0202. And as you can see here, that's the vena cava inferior prepared. There is no clot inside anymore. And these are the weight of the thrombi, and you can see in the majority of animals, the clot weight is significantly reduced. As I said already, we are dealing also with proteins which can be used in the treatment of osteoporotic patients. You know, osteoporosis treatment is sometimes limited for the time where you can apply the therapy. We are testing also drugs which can be used for targeting of multi-resistance bacteria, and this works also quite successful. And therefore, that is our main task, and that's what we are currently planning. That's the University Hospital of Mannheim here in this area, and over the street there will be up the new technical facility, 3 buildings in total. And in this floor, we're going to have our production facility, 300 square meters of clean room to do the GMP manufacturing for GLP tox, and also for the first clinical material. this will be a multi-purpose, multi-campaign facility to check new plasma protein candidates. We can optimize in this facility also existing processes and scale it up. And so we can use and also for the establishing of for the validation of single-use equipment, currently with a fractionation capacity of about 500 liters. We're going to hope to get it accepted by the FDA and EMA soon, and we have a couple of different cooperation partners. That's the current draft of the room planning with the analytical lab immediately included, clean room D and clean room C, because if we are planning to work here with single-use equipment in a closed systems, together with a cooling unit beside. And that's something what we can offer and what we suggest to do those local fractionation facilities, or to build up those local fractionation facilities in Asia to manufacture locally the plasma-derived medicinal products. Also, again, as multi-purpose, multi-campaign equipment. And the issue is that we can do this with the single-use equipment with up to 5,000 liters a week. That means that the small mini fractionation facility has a capacity of about 250,000 liters per year, which is already a lot. And if you're thinking about big countries, they can have 5 or 10 of them, so that they can end up with 10, also with 2.5 million liters. And we are establishing this just now here in cooperation with Merck, Biorad, 3M, PAL, and Asahi, and other groups, so that you can use this as a pilot plant, what we have just now here. This equipment looks a little bit like this. You know, and it's much smaller than the stainless steel facilities. You can avoid all the cleaning process, validation stuff, and a lot of cost. And that's a kind of example process, starting with a cryoprecipitate over here. Do the filtration with the single-use equipment, the solubilization of the filtrate. Chromatography can be done by single-use equipment, viral filtration, inactivation, SD treatment as well. And filling and lyophilization is something what we are planning currently to do this with contracted partners. So that is a kind of potential we have at Previe Pharma. We can do the process optimization according on the clinical demand. We can check also new candidates and we can bring them into production. And we can do also routine manufacturing, especially for small amounts, so everything is possible like this. So I would like to thank you for your attention, and in the case of contact, just give us a call or send us a mail. Thank you very much.
Thank you, Stefan, for this wonderful presentation. And I guess this speaks that how much work has already happened in this field, but how much more has to happen in the field of plasma fractionation. And I think the slides which you have presented is very, very impressive. And it also shows that how different sustainable models can be created within the plasma fractionation industry, because the Asian context or the Latin America and Africa may really not be the same. So probably we will need to look at more tailor-made models and work around it. So thank you very much for bringing it up. And I'm sure there will be a lot of questions, but could I please request you that we can request Zeev to present his talk and then we can take question if that is okay with you, Stephen?
Sure, I'm fine.
All right, thank you, Stephen, for this talk. Thank you very much. And in fact, I was talking to Mark just last month when I was in India, and he has a great plan. So please convey my thanks to him as well.
I'm going to forward this. Thanks a lot.
Thank you. Thank you. Zeev. Now I'm presenting Zeev. He comes from Israel and he has done a very good work and developed a new product. And so Zeev, floor is for you.
Thank you very much. Thank you.
Stephen, can you unshare the screen? Yeah, great.
Yeah, I need to share my screen if it's okay.
Right.
Great, great, great. So again, thank you very much, Ranjit. Pleasure to be here. Stefan, thank you very much also for the nice presentation. So I'm going to talk about that it's a medical device startup located in Israel. We had Ranjit few years ago until I hope my screen is now available.
Yes, it is very much available.
Okay, so now you can see it. So
Yes.
our focus, I hope you can see that the slides are changing. So our focus is in blood and in bleeding in general. As you know, bleeding is a major problem worldwide. We are aware of a lot of complications that can cause bleeding if it's a postpartum hemorrhage, if it's a cardiac surgery that went wrong, or if a car accident or anything that can cause massive blood loss. Worldwide, we are suffering from more than 5 million people that die every year because of blood loss, and mainly because of bleeding. Now, bleeding mainly affects young population between 1 to 46. This is numbers from the US, but also it's relevant to Asia countries like India and others, that most of the people that died because of blood loss are very young. And those deaths can be prevented. And also, you know, there are a lot of rehabilitation need if the bleeding is severe. And bleeding can be because of a major trauma or also surgeries in the hospitals. Today, the most common treatment for those bleeding patients is plasma. Plasma products are very common as the first line of defense to stop the bleeding. You can see here numbers from Europe from 2017 that overall more than 2.7 million units of plasma are being given annually in Western Europe. Those plasma units are being used mainly to provide a better clinical care for those patients and to stop the bleeding. But unfortunately, you can see here in the left part of the slide, a lot of plasma products has very low ability to stop the bleeding. And this is one article that demonstrates why plasma has very low ability to be a precursor for clotting and to prevent additional blood loss. This is the main reason why.
Steve, I just wanted to interrupt here. Are you moving the slides? At least I am not able to see the movement of slide.
Same here, Ziv.
No,
Same
even
here.
we can't see.
So
We also
Ziv,
can't.
just see that, you know, your slides. Yeah, you can restart it. Unfortunately, I wasn't able to see the movement of slides. So just want to make sure.
So maybe I will try again to upload. I don't know why.
Yeah, please do. Sure, please do that.
So I hope now you can see it.
I can see, yes, now I can see the slide number 4, I guess. Is that correct?
Yes, okay. Okay. Let me know if it doesn't move, okay, Ranjit?
Ziv, you may want to put it on the screen mode slideshow because, yeah, now it is on the slide mode. Perfect.
Is now you see the next slide?
Yes, yes, now I can see the movement. Perfect. This is going good.
Okay.
Thank
So
you.
as I mentioned, in Europe, we're talking about more than 20 million units of plasma products, blood products are given annually, but we're talking about 2.7 million units of plasma that are being given. As Stefan mentioned, and I'm aware of that, on, for example, in February 17, a few days ago, American Red Cross is now offering patients with financial compensation just to donate plasma. There is a world shortage in plasma product, and also in plasma as a bag to treat those bleeding patients. And we know that using solvent detergent or other treatment that try to remove any pathogen from the plasma can reduce its ability to be a precursor for to stop bleeding and to generate stable clots. And this is a phenomenon is well known, as this is published by the Octopharma, for example. So Plasma is trying to, and our mission is to try to give better care for those bleeding patients. If it's a cardiac surgery patient, if it's a trauma patient, by our single-use device, Clear Plasma. So we developed this cartridge, this cartridge named Clear Plasma. It's a single-use cartridge that can be connected by universal connection to any plasma donor. That means that we are connecting to the plasma donor that's supposed to go to the patient, and instead going with the regular line of transfusion, we are taking the plasma through the device, through the cartridge, And by doing so, we are removing unnecessary protein that interrupt with the coagulation system. By doing so, we are allowing to generate more stable clots that prevent blood loss and stabilize the patient faster. Our technology is a single use, of course. It's a non-pyrogenic, sterile, and very cost-effective technology that can stabilize the patient. Also, we have a very good patent on that protect on not only the device itself that filters the plasma, but also the output. That means that the plasma that we are plasminogen depleted plasma, we are calling that is abolished from plasminogen. So what exactly the cartridge is absorbing? Plasminogen mainly. As you can see, this is the results for more than 30 units of plasma used in our clinical study, pilot clinical study. You can see here that we are moving more than 90% of the amount of plasminogen, but in contrast, all other coagulation parameters, PT, PTT, fibrinogen, D-dimer, factor VIII, remain with no any change at all. That means that we are just removing from the plasma plasminogen and also 25% of the TPA, that is the precursor for plasminogen. By doing so, we are allowing the stabilization of the clots. Preclinical studies in animals, we demonstrated that we can minimize blood loss. You can see here preclinical studies in mice, more than 80 mice were used. You can see here that compared to regular plasma and also saline, we stabilize clots and prevent additional blood loss of more than 55%. In swine model, more than 28 pigs were participating in this study. We demonstrate that we can reduce bleeding in more than 55%, and time of bleeding was also reduced. This is our two clinical parameters that make the change between a patient that is being held in the emergency room to a patient that can go to the department. After successful preclinical studies, we went to the FDA to try to plunder the road to clinical approval in the US. And FDA recommendation was that to do 2 clinical studies with the clear plasma. So we are done 1 clinical study on emergency situation, that is upper gastrointestinal bleeding, and another clinical study that now is ongoing on cardiac surgeries. In both those indications, we are suffering from severe massive blood loss. Those patients coming to the emergency room or in the operation room suffer from severe massive blood loss that require plasma transfusion. And they are being treated more severely with any medication or treatment that can stop the bleeding. So we started to do the clinical study program with the indication of the upper GI. You can see that the length of stay in the US is about 10 days, and this is the cost of stay. So we did a pilot study on 7 patients to show safety. This study was done in Israel and also in Italy. Overall, we follow up the patient for more than 6 months to show that there is no any risk for thromboembolic or no any problem with that. We demonstrate, of course, that the device is safe, the plasma that we are generating is safe, and it reduced the time of bleeding like we saw in the animal, decreased number of platelets that needed, and reduced hospitalization time. This is the DSMB committee that was monitoring the study. All of them are hematologists from Israel and from Italy that approved the result. Following this successful pilot study, we did additional study that was on 50 patients. It was double-blind randomized control patient. That means neither the physician, neither the patient knew which kind of plasma they are getting. And that was done to show the device, of course, safetiness, but also performance to show that we can reduce, for example, the use of red blood cells, plasma, the hemoglobin drop, the consumption of platelets, and rebleeding episodes. And of course, length in the hospitals and mortality. The study overall took us 8 months. This is the site that were participating in this study, that including Israel, 3 sites in Israel, 3 sites in Czech Republic, and 1 site in Italy that Professor Franceschi was the PI of the whole study. Now, the results of this study show, first of all, that the clear plasma and plasma met the first endpoint We reduce any severe or major adverse event that can be related to plasma transfusion or to rebleeding episodes or to bleeding in general because of upper gastrointestinal bleeding. We had 0 mortality event in the tested group. That means that in the control group, in the people that receive regular plasma, we had 3 mortality events, unfortunately, of course. 2 of them was during the 30 days follow-up. 1 of them was in day 33. And this very similar to what is published in the literature, that between 8 to 10% of the patients suffer from death and mortality because of upper gastrointestinal bleeding, and we had 0. That means that we can minimize mortality using the clear plasma kit. In addition, as you know, in some cases when plasma transfusion is being given, there is rash and other side effects that can be used. In our technology, we demonstrated that there is no any transfusion-related adverse event or no any problem using our technology at all. On top of that, we demonstrated some efficacy results that, first of all and above all, we demonstrated that the biological effect that we are claiming is really active and also in human, not only in small animals like rodents and pigs. This is manifested by the D-dimer results. D-dimer is a biological marker that shows stability of the clots. You can see here in the point number 1 is that using our technology, we are minimizing the D-dimer. That means that the clots are more firm, more stable, and prevent additional blood loss. Once the patient were more stabilized and blood loss was minimized, they can be discharged earlier from the hospital. We demonstrated 70% reduction in hospitalization duration, and that was significant in all sites. Please remember that this study was done in 3 countries, in 7 sites, so it's still significant results. It's very interesting. Of course, blood product transfusion was also minimized. The number of platelets and plasma was minimized, but the most important thing is the rebleeding. Rebleeding, it means that the patient was discharged after it was treated. It can be between 2 to 7 days, but he returned back with the same indication to the emergency room. And in that, we saw that we had only 1 case in the clear plasma group, comparison to 7 with different severity in the tested in the control group. It means that the clots, as I mentioned, that we generated with the clear plasma were more firm, more resistant to lysis, and prevent additional blood loss. When we look on subgroup on patients that did not receive red blood cells at all, and still we evaluate the hemoglobin level, we saw that in the tested group, if you give only plasma to those patients, you see that the plasma increased the hemoglobin a little bit, but after that, the clots are being lysis, maybe a little bit, and we saw again drop in the hemoglobin. In contrast, in our group, even though the patient were in much severe condition, this is due to the randomization, but once you give him only the clear plasma, you can stabilize the clots and prevent additional blood loss up to 48 hours after that. As you can see, the differences is much significant in the clear plasma in comparison to the regular plasma. In these days, we are doing multi-central study on cardiac surgeries, enrolling more than 200 patients with bypass surgeries or valve replacement. As you know, this indication is very common. It's been more than 1 million operations are being done in the US alone, and also 1.5, I think, in Europe and China. And it's a very big indication. Still, the need is very, very high. More than 23% of the patients suffer from significant blood loss. And in this date, we're doing additional clinical study in these electives, clinical studies in Israel, in Poland, in Czech Republic, Germany, and also we're waiting for 2 sites in the US to be open after FDA approval. So again, in this study, we're going to compare between regular plasma to filtrated plasma in those 4 groups of patients. 50 patients that we will receive regular plasma in the cardiac surgery, in the bypass, 50 will get filtrated plasma in the bypass, and in the valve replacement similar. All in all, we enrolled 20 patients so far in several sites very successfully, and you can find more information here in the clinical group. If I'm summarizing the results of the 2 studies that were completed and now the ongoing study that is being run, we are getting very good feedback from physicians about the simplicity of the technology. The main point is that we are not changing any clinical practice. If you are using a plasma bag, you just can connect a device with a spike to the plasma bag, and that can go directly to the patient vein. We don't change time of transfusion. We are not reducing the time that you need. We are not affecting the need for more staff. And of course, it's very, very intuitive and very easy to use. On top of that, we demonstrated that in comparison to other technologies, our safetiness is above all. We are showing that we are stabilizing the clots formation. We are not generating new clots, and that is the difference between other technologies. We are preserving hemoglobin levels that allows the stabilization of the patient faster and to be discharged very early. We are classified as Class 2B. We are, I think, 3 or 2 months before being approved in Europe. We submitted the technical file already in July 2020. This is our last validation that we need to complete. That's going to be until April 17. Hopefully, then we will be certificate and can soft launch in Europe. According to the FDA, because there is no any predicate for technology like ours, we are classified as a PMA. That's good for us that there is no any similar technology available. And we are working, as I mentioned, to get the approval of the FDA to do the clinical study in 2 or 3 sites in the US. As you know, it's a huge market opportunity. Plasma and plasma shortage is very common worldwide. In Southeast Asia, I'm the very interested call with some of my colleagues in South Korea. So everybody is suffering also from Israel. We are suffering from shortage in plasma in UK and other countries, but still it's a very big market, very big segment. We have some competitors and this is the landscape of competitors that we are facing. For example, for Johnson & Johnson, they are having the Evucel that can support a patient with massive blood loss, but it's a biological glue that can be localized and topical mainly. There is the hemospray of Cook Medical that is used for upper GI bleeding, but again, it's a powder and you need to close the wound before you can use it. Octoplast, as I mentioned, as I show you, it's not relevant for massive bleeding. It can be used for dehydration. And that's a very good drug for brain hemorrhage, but the cost is very, very high. You can see that. And again, platelets and plasma in Israel, in Europe are become, because of the shortage, global shortage, because of the extended need that there is, the cost become very, very high. And again, we are affordable. Novo7, as I mentioned, is a very good drug, but it's risk, it's thromboembolic event. So again, this is the side effects that we are trying to avoid. We are a B2B company. We are doing some activities with hospitals to open excellence centers in Germany, in Hannover Medical School, in Düsseldorf, in Bavaria, in Austria, and others, because we are a B2B company. We're working with the demand of physicians and increasing the accessibility and insurance attraction. Because of the unique technology of our device, we won the Horizon program, the EIC grant and equity from the European Investment Canonical. They award us 1.8 million euro grant that's supposed to bring us to soft launch and 2.1 million euro equity that can take us to commercialization. With me, with my team, I have my operation team, Samach, my marketing director, Ines, financial team, and regulatory and quality assurance. This is our board and director and advisory, Yossi Schwartz from Mount Sinai. He's responsible for transfusion in Mount Sinai for more than 25,000 units of plasma annually. Ziber Nari is responsible for liver disease. George Alexander is helping us with trauma issues. That is a very big issue in the US. And Galia is a hematology that support us. Giora, Gil, and Land, and then from the US, all of them working us on the cardiac surgeries, opening us more sites and helping us with the relation with the FDA. This is our board of directors, including a representative of Asai Kasai, one of the biggest plasma filtration company. They're having a lot of filtration kits and filtration devices for plasma and for other blood products, and supporting us with progression and commercialization. Thank you very much. Thank you very special. Thank you for Dr. Ranjit for inviting me, and I'm hoping to answer any question.
Very impressive, Zeev. It was a very, very impressive product as well as the clinical data. It's very, very compelling data. And I guess when I was there with you, maybe August of 17, and sorry, 18, and we were talking of it that we have to start the clinical trial, etc. And we are seeing it is happening. So it's a very, very good progress in the last 4 years. So congratulations to you and your entire team. Very good work, and I'm sure people will have a lot of questions to ask you, and it's a great work done. So thank you, Zeev, for making it possible and sharing this unique technology with us. And we hope Asian countries and the other countries where this product can be used and save millions of lives. So thank you again. Before we get on to the question, I would request if anyone has a question, can directly ask or put it in chat box before we start the question answer session. Anybody has any question, please come forward. Please unmute yourself and ask the question.
I'm mute? No, I'm not mute. I'm here.
Any questions from the audience?
Yeah, Dr. Ranjit, I have one question with Dr. Stephen.
Sure.
Yeah. This is related to the quality control of the plasma derived product, especially in the case of IgG. When we are testing NTA, NTB, many a time we observe the hemolysis. So what may be the possible region for hemolysis?
Sorry, I have to unmute myself. It is known that there are antibodies against.
I can repeat. May I repeat?
No, I'm fine.
Okay.
It is known that there are antibodies against blood type A and blood type B present in the human immunoglobulin. It's not quite clear where they are coming from. These isoagglutinins are normally belong to the IgM type. But it seems to be that some of the donors had either contact with cross-reactive bacteria surfaces or stuff like this. So in very few donors, we find antibodies against these two blood types A and B. And so they are also in the immunoglobulin. And just now that was also developed by Brevi Pharma and together with Merck, that we had an absorbance which you can use in the polishing step of immunoglobulins to get rid of those antibodies. The structure of this absorbent is according to the carbohydrate structure of the blood type A and blood type B blood group antigens. And so by using this absorbent in a column, you can get those antibodies out of the IgG product. And the majority of the US and European plasma fractionators are using this absorbent already, due to the fact that are only very small amounts of those antibodies present. You need also a very small column only. So that's not as expensive, but it was a requirement from the FDA and from the EMA to make sure that those antibodies are not present anymore in the IVIG products.
Okay, thank you. Thank you, Dr. Stephen. Thank you.
Yeah. If you send me an email, I can forward this information about this product to you.
Okay. Thank you very much.
I will connect Dr. Prasad with Stephen, and probably, you can exchange the notes and see how can we really work on it. So thank you. Any other question? All right. I'm not able to see any question. Steve, sorry, Sangeeta. Yes, please go ahead.
It's just a comment for Zeev's presentation. It was such a good product that we came to know today through this platform. And the properties that Plus-Free has, I was really impressed on it with the type of, the D-dimers reducing, the reduction of the blood products usage, the reduction in rebleeding, so many of the things that Zeev has shown into his, I mean, the Plus Free has the properties. I found it very useful in the coming times, and I wish best wishes to the team and hope we see this product in the coming times soon. Thank you so much, Zeev, for that.
Yes, Sangeeta, we wanted to do some work with this product in India also. Only I wanted to, Zeev forgot to put my picture on his slides. My name is there along with the patent with Zeev.
Okay, good. No, but many congratulations for this product.
No,
Many congratulations.
yeah, no, he has done a very good work. I was in his lab. It was like an incubator center to coming to this far. It was a very small lab when I was visiting him, and it was like incubator center, maybe like 5-6 companies. Am I correct?
Yes.
Right, and coming this far is a really, really very hard work, and a lot of effort would have gone into it. Where Zeev is
Right.
so
Right.
when Zeev is in India, we will ask for a big treat. Yes. Yeah. So anybody, any question? Yes, Kalyan, please go ahead. Kalyan, unmute yourself.
Thank you, Dr. Ranjit. I have a question to Dr. Stephen. It was a really nice presentation, Dr. Stephen. And my question is that, can we produce PDMPs from the surplus plasma?
PDF, plasma derived?
Yeah.
I missed the last 2 letters. PDMCs, plasma derived medicine?
PDMPs, yeah. Plasma derived medical products. From the plasma that have been surplus in any blood bank. Like
yeah, sure.
we get the plasma almost 150 ml from the whole blood. So can we use that plasma for PDMPs?
Yeah, this plasma is named recovered plasma because of that's the plasma which is left after the red blood cell concentrates are produced. It's coming from leukodepleted red blood cell units, or you can do this without any leukodepletion. The positive part of this plasma is the fact that you have, due to the fact that those donors are coming in average in Germany about 2 times for blood donation. It's allowed for men to donate 6 times a year, for women 4 times a year, but the average donation frequency is close to 2. So these donors have a pretty high IgG level, and there is one but. The factor 8 is due to the fact that this plasma is taking up to 24 hours, and until it's prepared and frozen, the factor 8 level is pretty much down. So for the purification of coagulation factors, this plasma is not that much welcome, but any mil of plasma is welcome anyhow. But it's very good and successful done for IVIG and albumin. For 83 as well, protein C, it works also. So the majority of plasma-derived products can be produced from this recovered plasma. And as I said already, don't waste any mil of plasma, put as much as possible into the fractionation process. And I think for countries in Asia, the small scale single-use fractionation facilities, this is the future. Compared to the big factories, which costs hundreds of millions to get this established, to get this validated and all the stuff. And if you have once a small facility with single-use equipment, you can press the copy and paste button, and then it's done. So you have to validate this once and copy this for other regions with the same authority. So that makes life much easier.
Okay,
Yes.
thanks. Thank you, Stephen.
Yes, Stephen, you made a very valid point. And just to bring you what you said is a very, very important, that way back, I guess, maybe like 6, 7, or maybe 10 years ago, we did a study with the Indian plasma versus the plasma coming from the North Hemisphere. And we found the immunoglobulin level is fairly high in the plasma coming from Asia because of our various exposure to the public healthcare challenges. And it was as high as 38% more. And this was done on a large scale level. And we found the IVIG is more while the albumin remained more or less the same, but IVIG level was certainly more. So it's a very valid point in sense of the not only science and technology, but also from the point of view of the business. If you look at per gram of IVIG into international market, so it makes more scientifically and financially sustainable model in sense of the plasma fractionation.
Yeah, I'm absolutely agree with this. We compared also the European plasma versus US plasma, and we had in Europe about 10% more IgG than the US guys. And the reason is quite simple, and that's something what we have showed in the SIBLA trial. The IgG level depends a lot from the donation frequency. And so the conclusion in all the plasma centers I had it was that we are adjusting the donation frequency according to the IgG level of the donors. That has a huge impact on the fractionation when you can increase the IgG level in the plasma pool simply by donor guidance. So we excluded all the donors with low IgG levels, and we forced the donation frequency in all donors with a high IgG level.
Okay.
So that's kind of individual adjustment of the donation frequency, which is necessary and which has a huge scientific background.
Sure. Thank you. In fact, we have gone a step ahead, not maybe 10 years ago in my earlier assignment, where what we have done is, though it's very empirical work, we haven't conducted any clinical trial on that. But what we did, we used the plasma, the immunoglobulin, which is produced using the Indian plasma, and we given it to patients of let's say PID, etc. And we found that is more effective compared to the product which is coming from the plasma, which is non-Indian in nature. So it was an empirical data. We haven't done a systematic clinical study, but it shows, for example, if I'm using 20 vial for PID patient, but in this case, probably I was using 17 or 16 vial. So that was a huge, impact in sense of the patient care, if you look from the affordability point of view, because the antibodies probably were more matching on the account of clinical side. So this was also one of the studies which we did way back.
Yeah, also here I agree completely. In a local immunoglobulin, you have the local antibody spectrum against the local pathogens. And that's quite important. It would not be helpful. We have a situation partially in Europe that Danish plasma should be used only for Danish plasma recipients. That's a little bit too small. But if you're looking for a whole continent, and the area where people are normally traveling, that's important to cover this spectrum of pathogens, which should be covered by the respective immunoglobulins. And that's the next major reason for producing immunoglobulins locally.
That's right. That's right. Thank you. I think Satish has a question for Zeev. Satish, please go ahead.
Hi, Zeev. Thanks for the fantastic presentation. I would like to know what is the amount of plasminogen that can be removed from one cartridge, and how exactly you calculate how much plasminogen need to be depleted to see a positive effect, and what are the consequences to be expected?
Thank you very much for the question. So our cartridge has the capacity to remove up to 600 milliliters of plasma. It was designed like this because plasmapheresis is donors that are usually donated up to 600 milliliters of plasma. So it can be also integrated with a pheresis machine that we can remove more than 95% of the plasminogen. Now, as I mentioned, our capacity is up to 600 milliliters of plasma and more than 95% of the plasminogen concentration. Now, we found that it can impact the activity of the protein, of the enzyme inside of the body if you're giving 1.5 unit of plasma. That's tremendous because it's lowering your endogenous plasminogen. You understand that the patient had still its own plasminogen, right?
Mm-hmm.
We're just reducing its ability to lysis the clots, and by doing so, we are showing that the clots are more firm. As much as more plasma you give, of course, you dilute the plasminogen more and more, and you see it's abolished more and more. The effect is more strength. But you can see it from 1 and 1.5 units, you can see it right away.
Okay, thank you.
You're welcome.
Zeev, I have a question. When we were
Yeah.
doing the D-dimer testing, how
Yes.
consistent the results were in sense of the variability? Because the D-dimer is not a very straightforward test and it jumps all around, as my understanding. So
Yes.
how did you deal with such a difficult test?
This is exactly why we wrote D-dimer scope, because we normalized it from all of the sites in Europe and also in Israel.
All right.
It's very different, but still, we had the same trend, 4 hours, 12 hours, and 24 hours. We tried to introduce into the study the tag analysis, but
Okay.
not all sites has it.
Okay.
So it's a problem.
All right. Thank you. Thank you.
You're welcome.
So if there's no question and we are right on time, I would say that it was a wonderful presentation from both the speakers, Stephen and Zeev. And thank you very much for your contribution on Atom Forum. I sincerely appreciate coming on this forum and sharing your experience. And I'm sure many of us will get benefited with this. And as I see Stephen's work, I mean, Prairie Pharma work is going in very, very good direction on the account of creating more sustainable modular plasma fractionation models, which eventually would help countries like Asia and developing world. And I'm sure this will be of a great significant in time to come. And it's not only the financially, but technically these models will be more viable because typically these countries cannot collect millions of liters of plasma and make it work. So thank you very much, Stephen, and for making it possible. And I know last night I bothered you with my emails, etc. Sorry about it, but thank you very much for making it up. And Zeev, again, as a friend, thank you very much for taking effort and making this presentation. So thank you very much and wish you all the very best for the new set of clinical trial. And India is the next ground for you. So you're welcome.
Yes, yes.
Right. Thank you. Thank you, everyone. Sangeeta was not able to welcome all the guests. So I give her a chance to say a few words and then
No, I'm actually very grateful to Zeev and Stephen for making it and presenting such a wonderful we came to know about the new product plus this means the talk that Stephen had given, because as doctors in transfusion medicine, we actually are not much into the plasma field. And to gather this knowledge from this platform is actually very helpful. And from my side, as a chair of the PPT group, I'm actually very grateful and thankful to both of you. And of course, Ranjit, my co-chair, and my coordinator, Kalyan, they all have been so, they have always been present. And Dr. Prasad also is a part of our group. So thankful to each of you. And we look forward to see you in other forums also, prizing us with more details in all the plasma field that's coming up new. Thank you so much.
Sangeeta, we want you to stay in blood bank. We don't want to lose our jobs.
I would not come that side, don't worry.
We are with plasma.
Yes, Kalyan.
Yeah, thank you, sir. I think Professor Asadul Islam sir wants to comment something.
Sure, please go
Professor
ahead.
Asad.
Okay. Thank you, Ranjit, to again and again the program of the PPTA. Thanks, Sri Srikar. We are very knowledgeable. We learn more, and we are
Sure.
not in blood. Plasma is part of blood.
Yes, yes.
Blood contains plasma.
Sure,
So
sure,
it's
sure.
more. So I think Kollam rightly said, in an Asian country, if we work with the required plasma as little country, I think it will be very, very beneficial to us. If we to source plasma, it cost is very high and it will very, there is a legal issue. And I think we can more webinar, we know more knowledge and involve more people. And we extend people if we work together with the required plasma from our country, and we have to get pull them and get our center, factory center. I think little center. I think the Asian people will be more benefited because I am using albumin for my wife. It is very cost is very high. So if we work together and set up a plant here and with the
recovered plasma, at least some of the product we prepared and our people more benefited. Thanks the foreign speaker for their nice presentation. Thanks the Sangita and Ranjit, the other good friend, is working hard for this. Kollan is our, he's my son, he's my student, he's working. We are very happy. Within very short time, they are earning the Obina, and it will very beneficial. I think we have to use our students, our colleagues, so that they can get enrich their knowledge about the plasma products, plasma derivative, and others. In Bangladesh, we are trying with plasma slowly,
Thank you.
and in future we will try more. Thank you all. Thank you very much.
Thank you. I see Mr. Rajesh Verma from DCJI's office. Mr. Verma, would you like to add something here?
Thank you. Mani sir, and thank you so much. One question, because there is a lot of scarcity and of albumin and IVIG in India also, and price.
Mr. Verma, you are not
also
audible. Voice is going
model.
on and off.
So the label itself, the product can be available. Sorry.
Mr. Rajesh, you're not audible. Your voice is cracking.
Mr. Verma, can I take your question offline and forward it to Mr. Dr. Stephen and Zeev? I think he is not able to connect audio-wise. So I will take his question and pass it on to Zeev or Stephen, to whomsoever it is relevant. So thank you very much, and Kalyan.
Thank you, sir.
Thank you.
Thank you. So I have a question to Dr. Zeev. The product you have, showed to us, does it have any role in the recovered plasma?
Yeah, so we are trying to talk with big plasma fractionator and big plasma collection like CSL, because our vision is that any plasma that is being given to blood bank or hospitals will be already filtrated. As you know, for example, a lot of countries in Europe are buying from Octopharm or from CSL Behring, from Grifols, plasma ready for patient. And we are trying to convince them that using our technology, they can make plasma filtrated, safe, with lyophilized, with solvent detergent, but without plasminogen. That can improve clinical outcome in any situation that they will use it. So we are on it.
Thank you. Hello.
Yes,
thank you.
yes, Mr. Verma is online. Yes, please go ahead.
Yes, sir. Especially see the Indian scenario, like there, especially for the remote areas where it is very difficult to get the availability of the albumin and IVIG. So, can a small lab model can be developed by the company, so that it can be available to the patient also and to the blood bank also. So the blood bank also will get benefited with this.
Yes, this is a nice proposal.
I think probably Stephen may be able to take this question. Is it okay, Stephen?
I'm just now unmuted.
Right.
So you're absolutely right. The question is how to bring this IVIG to any region. And therefore, one of the opportunities is, as I suggested already, to build many small fractionations across the country with this single-use equipment. And the other opportunity in its early stage development is to go for chairside fractionation. It's micro scale fractionation to do this single unit by single unit that can be helpful as well. That's probably for countries like in Africa, where everything runs decentralized. When you are using even small mini fractionation facilities, you need to have a little bit of centralization, but you are talking just now here about completely decentralization. And that's something where the development should go really for chairside fractionation. And I think for products like albumin and RVIG, it should be possible, but that will be, I would expect, take a time frame of about 5 to 10 years at least before a process like this is accepted by the authorities, produced by the guys producing those apheresis machines and stuff like this. I'm thinking pretty a long time already about this, but I didn't find any of the plasmapheresis equipment manufacturers already who would be willing to do this.
Sure. I think, Stephen, just to add the final comment, I'm sure we are short the time, but just take another minute. What we are trying to do in India is I've been talking to few equipment manufacturers who are very keen into biosimilar and biopharma space, and they are very keen to bring, the plasma product availability and biosimilar, etc., in developing world. And they would be equally keen to participate and work alongside with you and Z both. And probably, it may be a more synergistic business model where there will be a mutually symbiotic and beneficial relationship. So maybe, I take Mr. Verma's question and I will also talk to him and figure out if something, we can do together with the companies within India, because we have a lot of good companies in India who are providing the very good quality water and tanks and so on and so forth. So can something be done on a more collaborative way? And I think that will be the way to go ahead to make certain things locally available with the technology and the local plasma. And that will make entire project more sustainable. So I think it's a very good point that Mr. Verma has raised from DCGI office. And I'm sure we will make another forum to have this discussion. So thank you, Mr. Verma, for taking this forward. I think I short the time.
Yes,
And I would request. Yeah, please go ahead.
Thank again because WCO is always emphasis to downstream plasma plant.
That's right. That's
So
right.
that is why I am raising this.
Sure. So maybe Dr. Kalyan, you can propose a vote of thanks.
Please for your required plasma. Collect what plasma with a good quality and we have future domestic plasma plant, at least albumin and other. It
Sure.
beneficial to your people.
Yes, definitely we can work
Thank you,
on it.
very nice proposal. Thank you.
Sure.
Thank you, sir. Thank you, Dr. Ranjit, for giving me the floor. And I think this is the time to place the vote of thanks. So good afternoon, everyone, to the one and all present here, and honorable chief guest, Dr. Ananda sir, Dr. Sangeeta, our scrupulous chair of the AATM Plasma Protein Working Group, and all the participants. It is such an honor for me to get the opportunity to thank all dignitaries, and on behalf of Asian Association of Transfusion Medicine Plasma Protein Working Group, I, Dr. Roy, the secretary of the group, extend a warm welcome to the people in the gathering. I would like to express my gratitude to all esteemed delegates of the webinar for their presence and contribution to make this webinar a great success. My hearty gratitude to the wonderful moderator, Dr. Ranjit Ajmani, for moderating the session in a magnificent way. My special gratitude and thanks to Dr. Stephen Kissing for his fabulous presentation on trends in innovation and development of PDMPs. Thank you for such an informative presentation, Dr. Stephen. And I extend my gratitude to our honorable guest speaker, Dr. Ziv, for giving us an overview of innovative blood filtration technology. Thank you for inspiring and encouraging us, Ziv. Both of your presentations were informative, engaging, and thought-provoking. We hope that the knowledge and insights gained from the webinar will be useful to all of you in personal and professional lives. My wholehearted appreciation to all the audiences who have joined from the different countries around the globe. Your participation is the true inspiration to us. And last but not the least, I'd like to thank Dr. Aikas Zindal for his endless technical support. Thank you, everyone, once again for making it a great success. And
Thank
have
you.
a great day.
Thank you. Just 5 second.
Thank you.
Just 5 second commercial from our forum. So we have another talk on 27 from
Right.
3 to 4 p.m. on Sri Lankan chapter. And that is where we're talking about the basics of plasma fractionation, right from plasma to the product and so on and so forth.
Thank
So you are welcome to join the forum. And we will send you the link on LinkedIn today and Facebook, etc. So you are welcome to join. And thank you very much once again, Zeev, Stephen, and
So
everyone.
one more ad.
Oh my God.
There's tomorrow a talk on gene therapy
Yeah.
on the Atom Forum, GJT.
Oh, is it so?
That's given by me. So please do come and join.
Yeah, please join. Okay. And
Yes,
we'll be
thank
really
you.
happy to
Thank
see
you.
you all. Thank you,
Thank you.
everyone. Have a nice
Thank
day.
Thank you so
Bye.
much. Bye-bye.
Thank you,
Bye,
everyone.
Doctor.
Bye-bye.
Thank you.
Bye.
Thank
Thanks
you.
a lot. Bye-bye.
Bye.